you so much, Professor Dr. Shiv Prakash. Very good overview on different kinds of fungal infections which are relevant to India as a species, the resistant to it, and clear call for research areas as well. Appreciate you sharing your knowledge and expertise on this area, which is really relevant to the Indian population. Moving on to one more relevant disease relevant to the Indian population as well, is about Leishmaniasis, and we have a real expert, Dr. Hira Nakhasi, who's going to be speaking on this topic. So Dr. Hira is the Director, Division of Emerging and Transfusion Transmitted Diseases in the Center for Biologics Evaluation and Research, which we know as CBER in the US FDA. And is currently responsible for approval of assays to screen blood donors for blood-borne pathogens and retroviral diagnostics to ensure blood safety in the United States. He is a trained biochemist with master's and PhD degrees from MS University of Baroda in India, and followed by his postdoctoral training at the National Institutes of Health, Bethesda, and even in Columbia University, New York. He's got tremendous scientific expertise, vast experience spanning over molecular virology, parasitology, cell biology, immunology, and vaccinology, with primary research focused on Leishmania pathogenesis and development of genetically modified Leishmania vaccines and diagnostic tools. With having an authorship of more than 170 scientific publications, including reviews and book chapters, he's also an elected fellow of American Society of Tropical Medicine and Hygiene and recipient of the multiple international prestigious grants from NIH, Wellcome Trust in the UK, and Japanese Global Health Agency. So with this, I invite you, Dr. Hira, to speak about India's R&D journey towards elimination of Leishmaniasis.
Oh boy, yeah. For short people, you need a step stool here. So thank you so much for the organizers for inviting me, and especially Dr. Vinod Mattu, whom I can. Huh? So how does it work? This is forward, this is backward. And I have to point it towards that? Okay. So, and also especially Dr. Vinod Mattu for inviting me, because we had been here for many other meetings, and when he talked to me, he said, we have been organizing this symposia here. Will you be able to come and present it? I said, I will be glad to do that. So obviously, so what I will tell you, the story which we have been developing over the last 25 years. And I'm glad Dr. Kavita Singh introduced Leishmaniasis quite a bit about it, so I'm not going to go into details. But what the story I'm going to tell you is not just me, it's a consortium of at least 13 or 14 groups which have gone from A to Z in the development of this vaccine, which is Leishmania vaccine. So as the introduction was already given, but the point I want to make from this thing is that the Leishmaniasis is not only now in the tropical and subtropical regions, but it is expanding into the areas which were not before there. For example, United States, which was recently designated by WHO that they as a cutaneous endemic for cutaneous Leishmaniasis. So, and in the areas where there are conflicts, these things are increasing very much too. So, but there are, you know, as it was mentioned, how many people, how many countries, how many deaths. I don't want to go into detail in the interest of time, but there are no vaccines available, and that is the important thing to eliminate. You can do all sorts of things, but at the end of the, as history tells us, vaccines are the ones which control the infection. And there are not really validated good diagnostic assays for surveillance and diagnosis. Now, so zeroing into India and neighboring countries, even though we heard that we are at the cusp of elimination of this disease, unfortunately, there are many, many cases, and as Dr. Kavita Singh said, that there are pockets where you can flare up, because what has been happening so far is that there, you know, you treat the patients or you spray the things, the sandflies go down, and the infection, stays for some time, goes down, and then after some time it comes up. So obviously it's time. And also, in addition to that, it is not the same species of parasites which are now affecting. There are changing these things. As you can see in Sri Lanka, it is this L. donovani which causes the visceral disease in in the India, now causes a cutaneous disease. Not only in Sri Lanka, in Kerala or in Himachal Pradesh, I was told, and in Bhutan, we recently went to Bhutan, and they think it is Leishmania donovani, which causes visceral disease, but it causes mucocutaneous there. So obviously, there are many facets of this disease. So the elimination of this disease, you need many steps. Obviously, you need active detection, effective treatment, control zoonotic reservoirs, vector control, education and communication, and the vaccine. And people have done that, quite a bit of that, but unfortunately we don't have every step a good solution to that. So therefore, I will focus on the vaccine part of it and also the diagnostic part of it, so that because it will be important for the surveillance purposes. So a few years back, there was a model developed how to eliminate if you need a vaccine, what would you need the efficacy of vaccine to eliminate the Leishmaniasis disease throughout the world in endemic area especially. And according to that model, which is the WHO's recommendation of 1 in 10,000 they consider it as a elimination achieved. So therefore, according to this model, if you have a vaccine which is 50% efficacious, it will take, to achieve that target, it will take at least 8 to 10 years to achieve the control. I don't call it elimination because I don't think we can eliminate anything. It is the control, and therefore, it will need a lot of doses of this vaccine. So with that aim, I just want to show you a few slides of what our efforts have been. It is not that others have not made an effort in there. Others have made, over the years, they have been developing these vaccines. Unfortunately, those vaccines never came to fruition. For example, they used the peptide vaccines, the DNA vaccines, and protein vaccines. None of them were effective. They were effective in the animal models, but when they went into the clinical trials, they didn't. The reason being that to understand that disease, you need to understand anybody who recovers from this infection, either it is cutaneous or visceral, is permanent, lifelong protected. That tells you that you develop a strong cellular immune response. So therefore, you need a vaccine which can develop a strong cellular response and therefore can protect you for a long time. So with that, we developed a genetically modified live attenuated parasite by deleting one of these genes called centrin gene. And in the interest of time, I'm not going to deal what the centrin is. It is important for the duplication of the parasite, but anyway, it is all published. So we developed this by CRISPR-Cas technology and showed that this candidate can be effective, doesn't cause any disease, and can be be safe and effective. And we showed in all those papers which we published that it is effective. And here and it is effective against several species. It's not just one species. This vaccine was developed using L. major, which is a dermotropic leishmania, and it can protect the visceral disease in the hamster model, and not only by injecting the parasite, but through natural infection, through sandfly infection, because that is the key. Sandfly infection, because all the previous studies which have been done for vaccine failed to prevent against the sandfly infection, because the sandfly carries many other components, not only just the parasite. So we showed that it can protect the visceral disease, it can protect the cutaneous disease in the mouse model, and in the dogs in Tunisia, we did studies over a period of 4 years where this vaccine was immunized to these dogs, and then every year they were taken to the endemic areas where the natural infection against sandfly was taking place. And after 4 years, when we analyzed the data, and it was more than 80% effectiveness. So this vaccine candidate told us that it can be a good candidate moving forward in the preclinical stage. Now, to achieve, then the challenge is how to move from lab to real world. So the target profile of this vaccine, of any Leishmania vaccine, has to have some of these characteristics. We put the fact together that it has to have, it should be protecting against various species, because it is very prohibitive, cost prohibitive to have vaccine for every infection, cutaneous, visceral, mucocutaneous, and all those things, or PKDL. So we won't put in sort of characteristics that it should protect against several species. It should be all endemic regions, target population, as well as travelers. It should be more than between 70 to 95% effective, have low adverse events, needs only one dose, not multiple doses, and it can be intradermal injection, lifelong protection, and then can be stored for a long time. And importantly, most importantly, where the vaccine is needed, has to be cost effective. So, which was less than $5, which was calculated at that time. So with the help of Genova, which this morning you heard Sanjay saying, it's not easy. The challenge is to find the manufacturer who can make it. We approached several manufacturers. You heard the names. I don't want to name the names today. The big companies, none of them took a bite because they think they are, what is it called? It is not cost effective. So we approached Genova, and they were kind enough to us to take this project upon themselves, and now they can grow up to 10 liters of this in the GMP facility, and you can see the vaccine is in the vials there. So now what are the challenges? So once we have a vaccine candidate, the next thing is to do the trials, and the trials are not easy to do, because if you do standard phase 3 trial, you can see the incidence of this disease varies over a period of time. And therefore, it will take years and years of that to do the effectiveness of that vaccine. So, and it is very difficult to do in certain areas where the cases are higher, such as in Sudan or other places. Those are very conflict areas, so you can do that. So how do you do then? What is the alternative to that using standard phase 3 trial? So there is a new model which has been, people have now used, it's called controlled human infectious model, or CHIMP model. This is just the attributes of CHIMP are, first of all, it requires a very small sample size in a controlled environment. It overcomes the uncontrolled fluctuation of the transmission, which I showed you, and then significantly reduce the cost. And the time frame, if you do standard phase 2, phase 3 trial, it will take at least 15 to 20 years to get the FDA approval. Whereas in the CHIMP studies, if you do, it takes 4 to 5 years. Because what you can do is once you have the phase 1 safety studies done and show that this vaccine is safe and effective, and safe, not effective yet, safe and a little bit immunogenic, then you can challenge those people with this challenge with the infection model. So we have, now also once you have that challenge achieved, so we are in the process of that. I will show you at the end that we have got the IND approval for it to do the phase 1 study. But in addition to that, now how do you monitor the efficacy of this vaccine? And the efficacy can be done by this simple test, which has been in centuries there, called Leishmania Skin Test, which is akin to tuberculin test. Or you can do an interferon gamma release assay. So again, Genova came to our rescue. They have now developed a field-deployable Leishmania antigen, which is in the wild, which is called, used for skin test. And we showed that in the preclinical and ex vivo human studies, that this antigen can induce a very strong DTH response as well as immune response. In the animal model here, we showed that it is both in the hamster and the mice model that once you immunize these animals and after some time you inject this antigen, you get a DTH response, delayed type of. In the studies in Brazil, where we did with the healthy active VL case, active CL cases and HEAL cases, we see a strong DTH response, as well as the interferon gamma IL-10 response. So, to summarize, where are we now with this vaccine development? We have the vaccine, with regard to vaccine, as I mentioned, we got the US FDA approval in July of 2025, and we have the funding to do the phase 1 studies in endemic areas in Kenya and Brazil, and non-endemic is in the US, so we will be starting sometime this month, or not this month, by March of these studies. With regard to this antigen test, which is both diagnostic as well as surveillance, we have now GMP material is being made, and I showed you some preclinical data that it works very well in the system. So to summarize, before I close my talk here, and I just want to acknowledge my partners in the crime here, and you can see that it is not just our lab in US FDA. Ohio State University, Dr. Satoskar is in the audience here, and you can address all the questions to him. And so, NIAID people, Genoa, Intox, Pasteur Institute in Tunisia, Nagasaki University in Japan, and our clinical partners there. And without the money, you cannot do anything. And we are thankful to Global Health Initiative Technology Fund, which have funded so far $15 to $16 million. NIH, Wellcome Trust, and of course my parent agency, the US FDA. Thank you very much.